Hello and welcome students. My name is Indranil and today I'm going to teach you how to design primers for mutating a few nucleotides in a sequence that we have cloned in PGL3 basic vector. So let's move on to the first slide. A brief introduction about PGL3 basic vector. So PGL3 basic vector is a mammalian vector which has some unique properties, like it does not have any eukaryotic promoter or enhancer sequences, which is usually required for transcription and all. It has this unique luciferase gene that is used for various applications with this particular plasmid. So it needs a functional promoter upstream of this luciferase positive gene. So this is a firefly luciferase gene. Upstream of it means in this MCS region, you need to have a promoter region which will help in the expression of this particular luciferase gene. So this particular vector is used for promoter luciferase assay, which is further used to validate transcription factor binding to the promoter of a particular gene. All right. So next I'll tell you a little bit about promoter luciferase assay, what it is. As you might have learned from the previous lecture, how to clone a particular, say, up to 1 kb region upstream of the transcription start site in a PGL3 basic vector, right? So what is promoter luciferase assay? This assay is used to confirm the or validate the binding of transcription factors to the promoter region of your gene. So for that, what needs to be done is the transcription factor binding site needs to be cloned upstream of a reporter gene. Here the reporter gene is your luciferase gene, and say you have cloned the transcription factor binding site upstream of it in that PGL3 basic vector which I had shown you earlier. Now obviously if your transcription factor comes and binds to the promoter region, the transcription will start and it will form the mRNA, which will further translate and form into the functional form of the luciferase enzyme, which upon acting on its substrate, it will give rise to luminescence, right, or light reaction. So based on this principle, if you say clone your transcription factor binding site and co-transfect that particular clone along with your transcription factor to mammalian cells, what will happen? Luciferase activity will be there, right? Luciferase activity will be there, and if you mutate the binding site of the transcription factor in the clone and co-transfect it with the transcription factor, there will be no luciferase activity. So just based on this particular principle, you do promoter luciferase assay, and there is this requirement for mutating a few sites, for example 3 to 4 nucleotides in your sequence. Okay, so moving on, mutagenesis. What is mutagenesis? Mutagenesis is a process by which you introduce some mutations in the nucleotide sequence of a particular organism, maybe, and there is some genetic alteration, right? So it can occur spontaneously or maybe by being introduced through some mutagenic agents like radiation, or it can be even be introduced artificially in the laboratory. So there are several techniques for introducing mutagenesis, like this, the kind of what we are trying to do, mutating three bases in a nucleotide sequence. There can be random mutagenesis, there is one site-directed mutagenesis, combinatorial mutagenesis, and many more. So what we are going to attempt is site-directed mutagenesis because it is a very precise in vitro technique in which you can precisely or specifically change the nucleotides in a particular region. All right. So this particular technique is an invaluable technique which is used to study the biological activity of various like RNA, DNA, even proteins also. So primer designing for SDM experiment is a very crucial step, and it is the initial step although, but I believe it is the most important step. If you do not design primers carefully, you are going to have much trouble while doing the experiments, right? So the mutagenic oligonucleotide primers for this protocol must be designed individually according to the desired mutation, right? So let's move on. So before I move on to the details of the primer designing, I'd like to give you a brief overview of how this particular experiment is performed, the site-directed mutagenesis experiment. So I would like to tell you that this is a PCR-based technique in which two overlapping primers are used. For example, let's look at this image on the left side. If you have, say this is your PGL3 vector, which you have cloned your transcription factor binding site or the promoter region, and here the transcription factor binds. Now you want to alter here two or three bases, and according to your wish, so that the transcription factor cannot come and bind here. All right. So what we need to do is, and for example, if you have these mutagenic primers in which at the center there is the altered basis, so what needs to be done first is you need to put up a PCR reaction with these two primers, which will do the amplification of this plasmid DNA. Now, what will happen here is, just look very carefully. After the amplification process, you will get something like this. Okay. Now, this particular amplification is done with a very good proofreading enzyme, for example, fusion polymerase, which has a very low error rate. Because we need to use a highly efficient and proofreading enzyme, because so that the unwanted mutations are not introduced. So upon doing PCR with these two mutagenic primers, you'll get a nicked circular kind of plasmid. And also, what is not mentioned here, this original, so this is a wild type plasmid. Okay, by wild type I mean the base pair here is unchanged. Now, after you do PCR with this mutagenic primers, the nicked circular plasmid that you get will have these mutations introduced in the center, right? This and this. But if you look very carefully, there is a nick here, here, and here. These nicks cannot be repaired by normal PCR only. These nicks can only be repaired by the host E. coli, I mean competent E. coli cells. So I'll come to that step later. So after you do your PCR reaction, you'll get a nicked circular, all and also along with this host plasmid, which has covalently closed double-stranded and supercoiled form. Okay, so this host plasmid does not have the mutation. This is wild type, and these mutations are also introduced here, so this is mutated. Okay, now what do we do? Now comes the most important step here. You will learn about a particular enzyme called DPN1. What is DPN1? DPN1 is an enzyme, that restriction enzyme, that only digests DNA which have a methylated site on it. Okay, so DPN1 only digests methylated or hemimethylated DNA. Now you might be wondering, I have written here plasmid DNA isolated from a DAM positive E. coli strain. What does that mean? So DAM positive E. coli strain has its plasmids methylated. Okay, so your host plasmid or your wild type plasmid is isolated from a DAM positive E. coli strain. It has methylation sites. Now, then you are doing the amplification with your mutagenic primers, right? Like I explained over here. Then what you need to do is digest this amplified product, which is a mixture of your wild type and mutated plasmid with DPN1. What will this do? This will completely digest this particular wild type plasmid in the sites where where methylations are there. So this particular wild type plasmid will be broken down into small fragments. Okay, and your mutated plasmid, just because it has been amplified and the methylations are not copied, right? It does not have any methylation site, so your mutated plasmid will be spared by DPN1, and it will only remain in your reaction mix. So after you digest your amplified product with DPN1 and hit inactivate the DPN1, then what you need to do is transform this nicked mutated plasmid into competent E. coli cells. In the E. coli cells, the cells will heal this particular nick here, and they'll form a complete supercoiled, plasmid. And since the nicked circular plasmid is introduced in the transformation process, the number of colonies might be less, but the colonies that you get, provided that the DPN1 digestion has taken place efficiently and it has eradicated all the wild type methylated plasmids, the colonies that you get should be containing the plasmids which have this methyl, I mean, mutation introduced in them, right? So the next step after your transformation is you pick the colonies, do isolate plasmids from them, and send them for sequencing in order to confirm or verify whether your desired mutation has taken place or not. Okay, so now let's discuss the few rules of primer designing while doing a SDM experiment. The first rule is both the mutagenic primers must contain the desired mutation and they must bind to the opposite strands in the plasmid DNA in the same sequence. Now, the length of the primers should be 25 to 45 bases, which you might have noticed is different from the usual primer designing rule, which is that the primer length should be 18 to 24 nucleotides long. But why this is here, we'll get to know in the next slide. Next, the desired mutation should be in the middle of the primer. Okay, so this is very important. For example, if you want to change, for example, three nucleotides in a particular sequence, you need to alter those three nucleotides, and you should have flanking nucleotide regions 10 to 15 nucleotides on each sides. These flanking bases will be same as the your original wild type plasmid because you don't want to change them. You want to change the three nucleotides that is in between. So these three nucleotides that need to be changed is very crucial that they remain in the center of your sequence with an equal number of nucleotides on each side, which can vary from 10 to 15 or maybe even more. The primers should optimally have a GC content of 40% because GC bonds make the binding a little stable, right? So minimum GC content should be 40, although in some cases you might not be able to get 40%, which I'll give you an example in the next slide. Now, it's very important that your primer should be purified, right? The standard synthesis is scale is desalted primer synthesis. Now, desalted primers, you can also do SDM experiment. It is not impossible, but everything gets a little bit easier if you have your purified product. Now, it is important to keep the primer concentration in excess because this entire technique is based on your primers, right? So the primer concentration should be a little excess. I'll give you an example where you do the SDM experiment. I mean, you design the primers for your SDM experiment. How do you do that? For example, I'll show you. Let's say that this particular sequence from here to here is the transcription factor binding site of gene X. So you can assume that this particular sequence is from the promoter of gene X, right? The transcription factor, it can be anything like it can be HIF1 alpha, HIF, or something else maybe. So anyway, the transcription factor comes and binds to its binding site here, and the transcription starts. The expression of the downstream gene is induced. Now the aim of your experiment is to design a primer pair which will change this particular nucleotide sequences here, and it will make some nucleotides in which the transcription factor cannot bind and your transcription or induction of transcription does not take place. So let us mutate this particular GCA, these three nucleotides in your sequence. How to do that? I have told you that these these three nucleotides should be changed. How do we do it? I have taken exactly complementary of these three nucleotides. So for G you have C here, and for C you have G here, and for A you have T here. Now this particular primer, forward primer, will bind. It has the same sequence as this, so it will bind to the complementary strand to this strand, and this particular primer will bind to here this sequence, right? Now as you can see that these three bases are altered, these three bases will not bind, but you need to take on each side at least 12 to 15 bases which are exactly the same. These 12 to 15 bases on each side will help the primers to bind to the template, original template DNA. Right, so like we had mentioned earlier, if this is your plasmid DNA and this is your forward primer, it will have the mutation in the center. Similarly, if this is your reverse primer, it will have the mutation here in the center somewhere. Then you do your PCR and amplify this product where you get a nicked circular plasmid. Then you digest it with TPN1 to remove the original host plasmid, and then you transform it into E. coli cells to get colonies, right? So it does not have many things to do. You just need to follow a few things that say the starting and ending. If it has C or G, it is good for binding. And on each side, I have taken here say 17 bases on each side, but still you can see that the GC content was not 40%. We could have taken even more, but I chose to take like 17 bases on each side here. The TM is about 71 degree, which is not bad. I mean, it is possible to do. And if you can imagine that if you're taking like 15 or 17 bases on each side of your mutation side, the TM will automatically go up. Now, a few things you might have noticed here. There is a stretch of A here. In normal primer designing lessons, you might have learned that stretch of a particular single nucleotide more than 3 is not advised. But in this case, you cannot help it because the transcription factor binding site is flanked with these sequences and it is not your choice to change this. So you have to take it. And what are the other factors? Just take 15 to 17 nucleotides on each side. Try to keep this the starting and ending nucleotides at G or C because it promotes binding with triple bonding. Try to keep the GC content to for minimum 40%. The TM will rise to 72 degree, no problem. And the primer length, try to keep it between 15 to 45. I mean, sorry, uh, under 45 nucleotides long. So there you go. This is very simple, not very difficult, but you need to keep in mind that this primer pair, I mean, they have at least close to G 40% GC content. They will form self dimers, as you can imagine. They are perfectly complementary primers, but uh, cannot be helped. This particular process uses this particular principle only. It will open up and bind to your I mean to template DNA. Now, moving on. Okay, so there is transcription vector binding, and here is not. Moving on, I have just shown you the few PCR cycling conditions where, say, initial denaturation is there, then your cycling and in your cycles, 95 degree for 30 seconds, 50. This is the annealing temperature. Usually, it's kept 55 to 60 for uh, SDM reactions, and these and I mean extension temperature depends on your polymerase, and the speed of extension it also depends on your polymerase. Now you might have noticed that the number of cycles is usually low than 30 cycles. This is because if you introduce more cycles, it will just produce smearing in the product. So that might not be intended. Okay, so for point mutation, the number of cycles should be less. Single amino acid changes. Now, since we have three nucleotides, which is represents or maybe represent a codon, so we should ideally do 16 number of cycles. And for multiple amino acid insertions or deletions, you can do 18. Okay. So after amplification and digestion with DPN1, you transform them into competent E. coli cells. We had done it in E. coli DH5 alpha, which are super competent. Then you pick the colonies, like you pick 3-4 colonies from here, and you send them for Sanger sequencing. So this is how a sequencing result chromatogram looks like. This is the wild type. As I had shown you in the previous slide that there was GCA, and after you do your SDM experiment and do all this stuff, and I got it mutated to CGT, just like I explained it to you in the previous slide. So that's pretty much it. And regarding primer design, just follow the few steps that I had mentioned, or the few points that I had mentioned. Try to keep them in mind. And I think Garima has taught you how to clone up to 1000 bases upstream of your gene's transcription site. So from that sequence, in that sequence, for your assignment, you choose a particular stretch of sequence, you identify a hypothetical transcription factor binding site, and you design the primer pair for your SDM experiment. Okay, so all the best to you, and thank you. If you have any doubt, we'll have a doubt clearing session a few days later.
Hi. So this was about site-directed mutagenesis, and now we'll be moving on to random mutagenesis. So we'll be focusing on error-prone PCR. Mutations, as we all know, are changes in the DNA sequence of an organism, and the process of artificially and deliberately inserting mutations into a segment of DNA so as to change its sequence in the lab is known as in vitro mutagenesis. The mutations introduced into the DNA in this manner can be either point mutations, which are most common and involve substitutions of a single base, or they can be insertions and deletions, or shuffling and rearrangements of large DNA segments. For in vitro mutagenesis, there are two strategies that can be followed depending on your downstream requirements. So, one is site directed mutagenesis, and the other is random mutagenesis. Site-directed mutagenesis, as the name suggests, creates mutations at specific pre-decided locations in the sequence. So it is precise and targeted. Whereas if the nucleotide sequence is randomly altered at multiple sites throughout, irrespective of any specific location, it is termed as random mutagenesis. And today we would be focusing on random mutagenesis. The question arises that why would we want to randomly mutagenize any gene or any DNA sequence? So the reasons can be multiple. The most common is in the functional dissection of an unknown gene or an unknown DNA sequence. So if we have some sequence and we don't know its function, the best way is to create mutations in it so as to disrupt the sequence, which will eventually lead to a loss of function in that gene, and this loss of function can then be screened and the functionality of the sequence can be assigned. Otherwise, it is also very useful in directed evolution and protein engineering in order to generate enzymes or proteins with desired properties. So suppose we have a protein and we want to, let's say, decrease its temperature sensitivity or increase its catalysis rates. So we would choose that DNA sequence and we'll keep on randomly mutagenizing it and select for the protein that is showing our desired characteristics. So after multiple rounds of mutations and selections, we eventually get the protein which is showing us what we need. It is also useful for determining protein structure functional relationships by mutagenizing specific domains of the proteins and finding its effect on the functions. And even the regulatory regions in the genome can be targeted by random mutagenesis. For example, the promoter region. So the promoter region can be mutagenized, and after rounds of selection, we can find the promoter with increased strength that is useful in expression systems. Or alternatively, we can mutagenize regulatory regions to find whether this region is indeed targeting some downstream gene or not. So, talking about the benefits of the technique, so since it's a random mutagenesis, it is totally okay if we don't have any prior sequence information because we don't need to design specific mutagenic primers, which is what is observed in site-directed mutagenesis. So it's okay if we just have the flagging region, so we can use it for PCR or cloning. Also, this technique is quick, easy, and inexpensive, which mostly holds true for the use of chemical mutagens or uh, error-prone PCR, for which all the reagents are easily available in the lab. We just need to alter some reaction conditions. There are different methods of carrying out random mutagenesis, some of which are listed below. So, firstly, error-prone PCR, which depends on utilizing error-prone PCR reactions in order to introduce mutations into the DNA sequence. Similar to error-prone PCR, we have the rolling circle error-prone PCR, which utilizes the circular plasmid as a template as opposed to a linear DNA. Next, we have mutated strains, which are those strains of bacteria that are deficient in DNA repair pathways. So we clone our gene of interest into these strains and we allow them to divide over multiple rounds. Now, since they are defected in DNA repair pathways, they have a tendency to keep accumulating mutations into their genome as well as our gene of interest, which can then be isolated. So next and the most simplest form of random mutagenesis is the use of chemical mutagens. However, these chemicals are extremely dangerous and it is at most important to handle them with care since most of them are also carcinogenic. So examples include EMS, which is an alkylating agent, and nitrous acid, which is a deaminating agent. So both of these are capable of introducing random mutations throughout the target sequence. There are also ways of carrying out insertional mutagenesis by means of transposons, which are jumping genes. So they get integrated into multiple sites throughout the sequence and result in insertional mutagenesis. Polymerase chain reaction is a method of amplifying DNA sequences in vitro. So the reaction basically starts with a mix containing our template DNA, specific primers, DNTPs, and a thermostable DNA polymerase enzyme. And with sequential cycles of denaturation, annealing, and elongation, we achieve multiple copies of our initial template DNA. When the PCR method is utilized for the introduction of point mutations into the DNA sequence, it is known as error-prone PCR. Error-prone PCR basically relies on reducing the accuracy of the DNA polymerase used, and this can be done via two methods, either by altering the reaction conditions or by using specific error-prone polymerases in our reaction. And I'll be explaining this in a little more detail in the next slide. So before we begin, let me just explain to you how DNA polymerases function inside the cell. So as it is visible in the figure on the left, DNA polymerases consist of two domains. One is the polymerase domain and one is the 3' to 5' exonuclease domain, which is also known as the proofreading domain. So in normal conditions, when DNA replication is going on, the polymerase domain is responsible for polymerizing complementary bases against our template DNA one by one. So while DNA replication is a very accurate process, it is very normal that sometimes the polymerase may incorporate wrong non-complementary bases against the template DNA. So if these wrong bases are not removed, they might result in a mutation. So in order to prevent this, the polymerase has the proofreading domain. So when there is a non-complementary binding, there is a change in the geometry of the DNA such that the exonuclease domain gets activated. And since it is 3' to 5' exonuclease domain, it removes the last wrong base that has been added such that the polymerization continues error-free. So, as opposed to conventional PCR reactions, where we want our DNA to be replicated with utmost accuracy and we do not tolerate even a single error in the reaction, for error prone PCR, we want just the opposite. So, the best way is to use DNA polymerases that lack this 3' to 5' exonuclease domain or the proofreading domain such that the intrinsic property of introducing the errors of the DNA polymerases is used. So the most common example of a DNA polymerase lacking this exonuclease proofreading activity is Taq polymerase. So Taq polymerase is still used in the labs because its error rate is fairly low. So it incorporates bases one. It incorporates error in one base out of 10 to the power 4 or 10 to the power 5 bases. So, so that is why Taq polymerase is still suitable for applications wherein we want to accurately amplify DNA that is 1 kb or lesser in length. So since Taq polymerase does not have a very high error rate, it can still be used for error-prone PCR if we alter the reaction conditions, which I would be explaining in a little while. Alternatively, there are special enzymes that have been genetically engineered, which are known as mutazymes 1 or mutazyme 2. So they are followed in kit-based assays. So basically, mutazymes are exonuclease lacking TFU polymerases and are known to be very error prone in nature. So you can directly use these enzymes in your reaction and follow the manufacturer's instructions in order to get error rates as per your um, applications. Alternatively, we can change the reaction conditions to achieve high rates of mutagenesis. So this can be done. So the most common way is by using manganese chloride in the reaction. So manganese is known to be a mutagen as well as a carcinogen because it reduces the base pairing specificity in the reaction. So the wrong bases are easily incorporated and in a non-template dependent manner. Similarly, if we increase the magnesium concentration, also the same can be observed. So magnesium is a normal cofactor for the DNA polymerase enzyme and it acts as a catalyst at one particular concentration. However, if we increase the concentration of magnesium ions, again, non-complementary base pairing is stabilized, resulting in incorporation of multiple errors. Next is if we use an unbalanced amount of DNTPs, it has been observed that the DNTP which has been provided in excess, so is a higher probability of getting incorporated more and more errors are created with the bias towards that nucleotide. Additionally, if we use base analogs or if we increase the concentration of the Taq polymerase enzyme, this also increases the probability of elongation of misprimed termini. Similarly, if we alter the PCR cycle conditions by increasing the extension time or increasing the numbers of cycles that we use, there's a higher probability that all the primers that have somehow bounced non-specifically to the template, they have a higher chance of getting elongated and there's a greater chance of accumulating mutations. So this is just a brief overview of the protocol that is followed with respect to the concentration of the components to add in the reaction mix. So this on the left gives the concentrations that we use for a normal PCR, and on the right is the difference that we see when we have to carry out an error-prone PCR. So basically the magnesium chloride, as you can see in a normal PCR, it is used at a concentration of approximately 1.5 millimolar. Whereas if we want errors, we have the freedom to increase it to up to 7 millimolar, depending on how many mutations we want. And DNTPs, if we have to obtain, if we have to give a balanced concentration of all 4 DNTPs, we add it at a concentration of zero point 0.2 micromolar. Whereas if we want an unbalanced ratio, so we can add zero point 0.2 micromolar of DNTP, and the DNTP which has to be added in excess can be taken up to 1 micromolar also. Uh, Manganese chloride is one component which is not added in normal PCRs, but it is added in error prone PCR at a concentration of zero point 0.1 to zero point 0.5 millimolar MnCl2, depending on the mutation frequency that you want. So, zero point 0.3 millimolar of manganese chloride is shown to give 5 mutations per kb. So for the error prone PCR reaction, we'll just assemble all of these components in an MCT tube and add the enzyme at last, and then we'll put it in the thermocycler for the reaction to begin. It's just important to prepare MnCl2 fresh and just add it immediately prior to starting your reaction. So the next thing is, how do we control the mutation frequency? So the mutation frequency depends on the DNA polymerase error rate as well as the number of duplications. So essentially, if the number of duplications are more, it's obvious that the number of errors incorporated will also be more. So the factors on which the mutation frequency depends are the cycle number. As I said, if there are more cycles, the mutation frequency would be higher. The initial template concentration is also important. So if we have a lesser template concentration, it will undergo more duplications and result in greater number of errors. And the manganese concentration. So on the right, there's a graph that shows the concentration of manganese and the number of mutations that it causes per 1000 base pairs. So as you can see, as manganese concentration is increasing, the mutations per 1000 base pairs also increase. So in general, if you want lesser number of mutations, you should aim for higher template concentration that is still in the range, lower cycle numbers, and lower manganese chloride concentration. And on the other hand, if you want high mutation frequencies, you should take lower template concentration, higher cycle numbers, and higher MnCl2 concentration. So this is the protocol for using the mutazyme enzymes. So mutazyme 1, as I said, it is the exonuclease lacking PFU polymerase. So there has also been another variant that is developed, that is mutazyme 2, which is a blend of mutazyme 1 in addition to a Taq polymerase mutant. So mutazyme 2 is proposed to have higher error rate when compared to mutazyme 1. And if you want to read more details about the Mutazyme protocol, you may refer to the user manual, the link of which I have given below. So, yeah, so this is about the random mutagenesis. If you have any doubts, please feel free to ask me. Are there any doubts?
Hello, ma'am.
Hello.
Ma'am, I have a question about site-directed mutagenesis.
Yeah.
Actually, as it was mentioned that the primer concentration should be in excess and also the primer should be desalted. So when we check the primer properties in the gene runner, do we need to change the default parameters we generally use?
No. That has to be kept the same. The primer concentration the probe concentration that you keep at 10,000, that has to be kept the same.
And the salt concentration, I think we keep 25
I think,
millimolar.
yeah, that is, yeah, 25.
That will also be same.
Pardon?
That will also be kept same, 25 millimolar.
Yeah, that would also be kept same. So we don't need to change any of those.
Okay, then what is the meaning of desalted primers?
Okay, so when primers are made in the company, there are also additional salts in the reaction, and we don't want them. So it's just a way of purifying the primer phase, wherein small molecular weight salts are removed because we just want the pure nucleotide sequence. So that you would have to specify when you're ordering your plasmid. So you'll get an option of either using desalted or HPLC purified. That is the order that you place to the company. So to them, you need to specify that you'll aim for FPLC or HPLC grade purified primers.
Okay, thank you, ma'am.
Yeah. Okay. Any other doubts?
Hello. I had
Hi.
a doubt. I think I missed it during the class. I had a network issue. Anyway, the doubt was that during site direct mutagenesis, the two primers they are acting on the same sequence, just on the opposite strands, and
Yes.
they're just differing in just three nucleotides. So I was just
Yes.
wondering, wouldn't they be there be a very high chance of forming primer diamonds amongst themselves?
Yes, yes. So there will be a very high chance of forming primer dimers because obviously both of them are complementary. So that is why we need to add the primers in excess, as sir mentioned in the slide.
Okay. So to increase the chance that some of them might
Would
bind to our
eventually
target.
bind. Yes,
Okay.
yes.
Thank you.
Okay, any other doubts? Okay, so we'll move on to the next lecture and I'll be handing it over to Anuja. Anuja, are you here?
Yes, ma'am. I will just share
Okay,
my screen.
okay, okay.
Is it visible?
Yes, Anuja,
Is
it's visible.
okay. I'll just play it then. Hello, everyone. Hello, everyone. So this lecture is about designing primers for cloning CDS region of a gene with N-terminal his tag in the PET vector. So for this, first we need to retrieve the CDS region of our gene of interest. Here I've taken the example of MET30 gene. So following the previous lectures, you must be aware of how to actually do it using NCBI. PET vectors are basically bacterial expression vectors, which are used for cloning and in vivo expression of recombinant proteins in large quantities in bacteria, especially E. coli. And here we're using the example of PET28A vector for cloning. Now, since the PET28A vector already has an N-terminal his tag, we can use NDE1 or NHE1 as the restriction enzyme of our choice, along with any of the other restriction enzymes in the NCS region to clone our CDS. This will ensure that the T7 tag is removed and the fusion product that we have has our gene of interest along with the his tag. To select which restriction enzyme to use for cloning, we upload the sequence on NepCutter, and here we look at the list of zero cutters or unique cutters, and finally uh, select the specific restriction enzyme that we'll use for cloning. So without going into a lot of details as to how to design the primers, which I'm sure you must have already learned, here I've simply taken the example of the primers, and also I've taken NHE1 and HIN3 as the restriction enzymes that I'll be using. Uh, Once we have designed the primers, we need to check whether the CDS region that we'll be cloning that would be in frame with the his tag, which is already present in the vector or not. So for that, first we need to retrieve the sequence from the his tag till the NHE enzyme site. from the vector, and then we'll upload it on Snapgene to check whether it would be in frame with our CDS or not. So I have retrieved the sequence from the vector and I've shown it in green on the slide. So I have now uploaded the sequence that I retrieved from the vector along with my CDS on Snapgene, and using the translate option which has been shown in the toolbar here, I can see that my protein would be in frame with the histag. So sometimes it might be the case that the protein need not be in frame with the histag. In that particular case, we'd have to adjust the base pairs by adding a few additional bases that would make our CDS in frame with the histag. I hope that this lecture was clear to you all. In case you have any questions, you can let me know. Thank you. Hello. Hello. Was this clear to you all?
Hello, I just have one doubt. I might have missed it, and maybe it was explained in one of the previous lab sessions. So here, like in this case, there are lots of nucleotides between the N-terminal histag and our gene of interest or our region of interest. That is the CDS. So is that not a problem based on what exactly we want to do once we have cloned it and once the protein is expressed?
No, so that is generally not the problem because you might have noticed there's a thrombin side between the histag and our CDS region.
Right.
So that side is usually used to cleave out the histag. So histag, like tags, fusion tags, they're basically used to further downstream purification of our protein, so or other functional assays or other functions. So if we're using this histag and we have the thrombin side, we can cleave it out and it won't be a problem for our protein, the recombinant protein that we'll get.
Okay. All right. Thank you.
Any other question?
Hello.
Yeah.
So if we are cleaving the tag with the thrombin site, so do we need to clone our gene of interest with the start codon? Because if the ATG is present in front of the histidine tag, so if we are cleaving it off, the start codon would also be cleaved off.
It's not very clear. Like you want to ask that the ATG is present.
Start, yes. Should we clone it with ATG? Because I have not added ATG. I thought ATG is already present in front of histidine tag.
Okay, so it actually doesn't really matter because ATG also codes for an amino acid, right? So ATG is also present in like if you see the CDS, so it can be not like it can function not just as a start codon but also it can code for the amino acid. So here I have incorporated it because it doesn't really matter uh, since it's already like the histag would already the translation would already start from the histag, but even if ATG is present in the sequence, it won't make any difference as such.
Okay,
Yeah.
thank you.
Is it clear to everyone? So if there are no other questions, we can close the session and you can let me know. You can write to me if you have any other questions. Okay, thank you everyone.
Thank you, ma'am.
Thank you.